welcome you once more to this Liver Seminar 7th edition session. First of all, I'd like to thank all the attendees for connecting to this session. Your presence here and participation is highly appreciated. Also, I'd like to thank the organizing committee for giving me the opportunity of hosting today. And of course, my special gratitude to Dr. Josep Maria Argemí for accepting the invitation. So I'd like to introduce to all the attendees Dr. Argemí with a brief summary of his impressive trajectory. He was graduated from med school in 2003 by University of Barcelona and then further board certification in internal medicine in 2010 by Biomedical Campus University of Rome in Italy. After that, he completed his PhD studies and thesis defense at the Center for Applied Medical Research, CIMA, that belongs to the University of Navarra, in 2015. So during his PhD studies, he focused on the role of stress response of misfolded proteins in post-hepatectomy liver regeneration, which resulted in a high-impact publication in Gastroenterology Journal in 2017. Soon after he obtained his PhD degree, he joined the pathology unit of the Clínica Universitaria Navarra, CUN, and from 2017 and 2020, while simultaneously he was completing a postdoctoral stay at the University of Pittsburgh for Dr. Ramon Bataler's lab, where he worked on the role of HNF4 in liver failure in patients with alcohol-induced hepatitis. So this postdoctoral stay was the moment where I had the opportunity to meet in person, work closely with him, which I deeply appreciated, and I thank him again for all that he taught to me. So since 2020, he has also been PI at CIMA, where he investigates the role of reticulum stress in the immunogenicity of hepatocellular carcinoma. And concomitantly, he carries out clinical duties at the CUN Liver Unit and also participates in clinical trials and teaches at CUN Med School. He has succeeded obtaining funding from several competitive national grants. And finally, he's a member of the Scientific Committee of Cancer Center and the Strategic Committee of the Center of Applied Medical Research Genomics at CUN. So again, thank you very much, Dr. Argemí, for joining us today and bringing this fascinating topic, which is about the role of the unfolded protein response in liver cancer immunogenicity. Please, Dr. Argemí, whenever you're ready, we're pleased to listen to the talk.
Okay, so thank you very much for inviting me. Thank you, Anna. Thank you, Rafa and Jordi. And thank to all the people is now connected to this topic, to this talk. I'm going to talk a little bit about the role of the unfolded protein response in liver cancer immunogenicity. And the outline will be this. I will talk briefly about hepatocellular carcinoma, which is the most common primary liver cancer, and its immunogenicity. And what is the endoplasmic reticulum stress and the response to this stress, which is called the unfolded protein response. Why we think that the UPR, so the unfolded protein response, could be a good target in HCC. And I'm going to talk also about our experimental setting here at CIMA, and then some preliminary results we have with this very recent setup research path that we have here in the lab about XBP1 and its role in the MHC class 1 exposition, and then some brief conclusions. So to start for the brief presentation about HCC, for those who are not familiar with the topic. So HCC is most common primary liver cancer, is the third leading cause of cancer-related deaths. And it can be classified in terms of the expansion of the tumor in the body in different stages. These are the stages that were defined in the Barcelona Liver Cancer Clinic group. which comes from the very early stage with the single nodules, solitary, small, and then if the tumor is a little bit larger or different, several small nodules, it's an A stage, and then from this stage on, the tumor is so big, or there are so many nodules that they cannot be applied for curative treatments, and they have to be treated with systemic treatments. In the stage B, you can combine with local regional treatment, but they are not considered fully curative. So most of the patients are diagnosed at very advanced stages, because it's mainly asymptomatic in the early stages. So the patients come to the clinic with symptoms and mostly are in a very advanced stage. And for what we know from models in animal models, cell lines, preclinical experiments, and human data, is that there are several molecular targets that are interesting and that have been historically blocked to try to fight this cancer in humans. We have growth factor receptors, it's called tyrosine kinases, EGFR, FGFR, VEGFR, PDGFR, EAT and MET mostly. And there are several drugs that have been developed for blocking these targets. Then we have the immune checkpoint system, the immune surveillance that is meant to kill the cancer cell, and it's blocked by the expression by cancer cells of PD-L1, that is the ligand of PD-1, and so it blocks the activation and killing activity of T effector cells, and this is of course another very important molecular target. And finally, we have also VEGFR, that it's not. It's mainly in the endothelial cells, although there is in other cell types, and it's also the third arm of this of the current systemic treatments for hepatocellular carcinoma. So this is a brief landscape of systemic treatments in hepatocellular carcinoma. So for many years, we have only the efficacy of sorafenib that started with the data of the SHARP trial. Then lenvatinib was not inferior. But then after many years, immunotherapy came also for liver cancer. It was already applied to different cancer types. And finally, we have a first phase 3 clinical trial that was positive for atezolizumab. And then other combinations have been also effective in phase 3 clinical trials. And there are upcoming results of different combinations, either combining 2 immune checkpoint inhibitors or combining 1 immune checkpoint inhibitor with a VEGF system inhibitor, or TKIs with checkpoint inhibitors. This is the backbone of all systemic treatments, and we are waiting these results to see which is the best combination in first line and also second line, etc. But the reality is this. So these results with only nivolumab, which was not superior to sorafenib, and so the nivolumab was not licensed for first line in hepatocellular carcinoma, but it is useful also for every immunotherapy. So we have some patients that are doing well, and for the first time we're seeing a lot of complete responses with immunotherapy, but this is true for only 1/3 of the patients. So there are many patients that are, let's say, maybe slow progressing or progressing or rapidly progressing under immunotherapy. So we need something else for making hepatocellular carcinoma a more immunogenic And this is as an example. So this is a patient from last year here at CUN, so at CUN. Sorry about the Spanish. I think this is a patient, 68 years old, with alcohol-related liver disease that had a very large tumor, as you can see here, and was not only large in size, but also invading the cava vein and going up to the right heart. And he was treated with under, in a clinical trial regime. And it had the monotherapy arm with nivolumab. And it has a very, as you can see with the AFP and also the images, has a huge and very rapid response to this immune checkpoint inhibitor, anti-PD-1. So that in a very brief time, the tumor was completely respondent. Now the patient is without any therapy, so it's completely cured of this hepatocellular carcinoma. It's something that is very new in the field, but so we are seeing certainly these kind of cases, but unfortunately most cases are like this other one. So this patient was under sorafenib for a lot of time, and the tumor was very slowly growing, and then as soon as it started progressing with meds in some spine bones, we switched to double immunotherapy, but it was not respondent. So we switched to another TKI like cabozantinib, but it was advancing. So we switched to atezolizumab. It was like stability for some time, but then finally the tumor growth so occupied the full liver, and the patient died. So this is and as you can see, for the survival, it was 2 large years, but it was a non-responder patient. So this patient has long survival because of the slow pace biology, but he was completely non-responder to the immunotherapy. So we need something to make this tumor more immunogenic. So that the mutations in some other cancers, we have target therapy. So we have driver genes that can be targeted because they are receptors or because they are kinases. In hepatocellular carcinoma, we have mostly 2 driver genes, beta-catenin and p53. None of them at this point are targetable, even if this one paper this last year about Wnt pathway inhibitors that could be useful for beta-catenin mutant tumors. But this is a reality. And then we have a very low tumor mutation burden in the patient. This is a TCGA. So overall, this is a tumor mutation burden related to other, let's say, more hot tumors. So and then this is the work of the group of Jovet, Daniela, Sia, and others. So it's clear that most tumors are not hot in terms of immune cell infiltration or immune cell signatures. And even those that are hot, sometimes they don't respond because the immune system is completely exhausted. This is an example of the complication of the complicated landscape that the immune system in HCC is. So this is a single cell on HCC and different stages. So this is TNM stages. So this is early stage. This is stage 2, and this is stage 3. And as you can see, sometimes it's difficult to see these graphs, but this is a trajectory UMAP, and the green are the patients, the cancers in the stage 2. The red are in stage 1, and the blue are stage 3. So you can see that the tumor evolves immunogenically as the tumor grows. So this is the pattern of cells, of immune cells in the tumor stage 1. Can you see my pointer? Yes. Okay. So these are the immune cells in the stage 1. They completely change when the tumor is growing, and then change again when the tumor is growing a little bit more. And the changes are related to the decrease of important markers of effector cells. For example, here, that it will be the tumors in stage 2 have lost granzyme B, and they are rich in PD-L1, so they are more immune evasive than before. They have lots. They have dots of FOXP3 cells, which are Tregs, and they have also other signs of immune exhaustion, like CTL4, that were not at the beginning. No, the beginning was this, and then they become rich in CTL4, and finally they. So the immune system is not fixed, so it's changing in terms depending on the evolution of the cancer itself. And then to complicate more the things, so the important thing, this is again the work by Daniela Sia, and the important is not just the immune system within the tumor, which is this one, but also in the surrounding tissues. So the immune signature in the surrounding tissues could be more defining of the immune response, of the immune checkpoint inhibitor response than the tumor itself. So this is very interesting and makes us think that probably we should investigate also the immune system in the surrounding liver, not just in the HCC. So basically, the challenge that we face is that we have to revert the immunosuppressive environment in HCC. This is so for this is the Clínica in Navarra, CUN. This is the CIMI, and we are trying to go back and forth, like you in Gregorio Marañón and clinic do the same, with bringing ideas from the vet and trying to make them, modeling them in the research center, and come back with some new targets and opportunities for our patients. So what is the endoplasmic reticulum stress and the folded protein response? So this is a cell. So we have the nucleus, nucleoli, and then we have the endoplasmic reticulum. This is the rough, this is the smooth, and then we have the Golgi system and the membrane. So basically, when an mRNA is translated by ribosomes, it enters the ER in an unfolded form, and the ER is devoted to the folding of these proteins, so that they can make their proper function. So you know that many mutations, just changing a loop in the tertiary structure of the protein can completely impair the function of it. So it's very important that this system is very well tuned, and so there is a whole pathway in the cell that takes care of this, takes care of the proper folding, and if something is happening with a certain protein, it gets retrotranslocated to the cytoplasm and get degraded by the proteasome system, and if not, it can go to the Golgi system and then secreted as a secreted protein, or incorporated to the membrane as a transmembrane protein. So how is this regulated? So how the proteins can be regularly folded? So I'm not going to talk about the intricacies of the mechanism of protein folding within the ER, even if it's super interesting. But I'm going to talk about what happens when the proteins do not fold correctly. And this can happen in oxidative stress. It can happen with, there is a lot of overload of the endoplasmic reticulum, or there is some other types of stress, like malnourishment, or is it hypoxia, etc. So that the proteins, there is not enough energy in the cell. So some proteins cannot be correctly folded. So then what happens? So these three receptor systems are activated. So ATF6 is a transmembrane protein, which luminal domain, cytoplasmic domain. So when there is ER stress, so unfolded proteins, it gets translocated to the Golgi. And then there are some proteases, S1P and S2P, that can cleavage the cytoplasmic domain. And this cytoplasmic domain is a very active transcription factor that goes to the nucleus, start synthesizing UPR target genes, which normally are chaperones, for example, that help the misfolded proteins to fold better. There are also genes that are related with the endoplasmic reticulum related degradation. Or there are other proteins related with the tuning of the response to hypoxia, et cetera. So the same happens with PERK. PERK is a transmembrane protein, which is a kinase. This kinase, upon ER stress, they oligomerize and they trans-autophosphorylate. And then they phosphorylate the factor of initiation to alpha. So eukaryotic initiation factor to alpha. This is the most important factor for the translation of proteins in the cell. So at least one of the most important. And when there is phosphorylation of this factor, the global translation of the cell goes down. So there are less proteins entering to the ER. So it's a little bit relaxing this stress, is reducing this stress. And at the same time, there is a upregulation of the translation of a specific mRNA, which is ATF4, that goes to also is a transcription factor that goes to the nucleus and also activate response genes. So PERK phosphorylates AF2 alpha, reducing the protein translation globally in the cell, but increase the protein translation of specific mRNAs that have a specific structure in the sequence of these mRNAs to be translated in these circumstances. So then finally, we have IL-1. IL-1 is another kinase, transmembrane domain. When it detects the stress, it also oligomerizes. And what this protein does, it has a RNase domain, which basically is RNase that also has is coupled to a ligase function, so that when an mRNA that has a small intron in the sequence, that's normally this mRNA is all around the surface of the endoplasmic reticulum, so it doesn't have to travel here upon stress, but it's already coating the endoplasmic reticulum. So upon activation, it splices this small intron, and the ligation happens. And then this new RNA that has been spliced with this small intron here changes completely the open reading frame. So now it can translate for a very active transcription factor, which is XBP1. And so XBP1 goes to the nucleus and start synthesizing, increasing the expression of genes related to the response to this stress. So basically, this is the system, very summarized, and it has there's some other things that I have not explained that are also happening at the same time. For example, IL-1 can activate JAK kinases, can initiate the decay of some RNAs. So, and these pathways could lead to apoptosis instead of survival of the cell. So there's always these two phases of the UPR. So if the UPR is activated, normally the cell will survive. But it's chronically activated. Other pathways that depend also on these activators of these sensors will lead to apoptotic pathways. So the cell will die if the stress is chronically there and it is not solved. And what is the relationship with cancer? With many hypotheses and many lots of papers about ear stress and cancer, and it makes a lot of sense. So basically, there are a lot of circumstances in the tumor environment that are hostile, and also cancer therapy can be hostile to the cancer cells, obviously. So all these things in the cancer cell produce protein misfolding in the ER. So it leads to the activation of the ER responses and the UPR. And so then there are different consequences that are that the cancer cells can do in order to avoid the apoptotic side, the apoptotic phase of the UPR. For example, they induce a lot BIP, which is a survival chaperone, is the most abundant chaperone in the ER. We'll talk about that. It can downregulate p53. It can decrease a CHOP expression, which is the apoptotic target of ATF4. So it can promote survival. And so these cancer cells develop the capacity to survive under ER stress. Also, we have seen that MYC overexpression leads to increased autophagy, so it can reduce the ER stress that way. Same with the loss of PTEN that has different adaptation responses to cope with this stress and survive, and so be more resistant to the stress. So there is some reasons here already to think that the targeting this pathway could be a good thing in cancer. But I'm going to talk specifically about HCC. So basically, this is a liver cell. So liver cell is, as you can see in the cytoplasm, is fully, completely packed with endoplasmic reticulum. And then what is not endoplasmic reticulum is mitochondria, which is completely in a very intense relationship with the endoplasmic reticulum. So the ATP and the calcium is fluxing from these two organelles, so they need each other. So basically, it's a system that it's completely programmed to produce and secrete plasma proteins and tangible membrane proteins. So it has a lot of endoplasmic reticulum. So probably, epigenetically is ready to have a very hepatocellular carcinoma is ready to have a very good UPR to cope with stress. This is a very beautiful paper that shows very elegantly that if you this MOP UPA mice are mice that are expressing the plasminogen activator and their constitutive promoter. So it's a secreted protein, but it's expressed in so much amounts that there is a lot of ER stress in this animal model, in these cells. So basically, these researchers put these mice into low-fat diet or high-fat diet, and what they see is that the mice who had these hepatocytes with a lot of ER stress, so secreting this high amount of plasminogen activator, they had more severe steatohepatitis, more severe fibrosis, and there is a lot of ER stress in these cells, in these livers. And then there was more coming of HCC in these mice. And then if they treated with chemical chaperones, which reduces the ER stress, so the effect of this genotype was greatly reduced. So it means that ER stress itself, very purely produced like in this model, could lead to an oncogenic environment. And if you reduce the ER stress, you could reduce the amount of cancer in these models. and what happens in humans. So this is an evidence from 20 years ago, and they look. This is northern blot, so it's old-fashioned, but you can see that in many. This is non-tumoral, and this cancer, you can see that many cancers have high amount of GRP78, which is the BIP, is another name for BIP, which is the main target of XBP1 and ATF6. So all of them are overexpressing BIP, and they see here this is well differentiated, moderately differentiated, and poorly differentiated. So as the cancer is more aggressive and more poorly differentiated, it has higher ER stress. So it was very interesting. And then there are many papers around these years about the presence of these markers. And as you know, HSP70 is a good marker, histological marker for hepatocellular carcinoma, precisely because it's a good marker of ER, and protein folding because it's cytoplasmic. Sorry, but what these researchers found is that also GRP78, which is an ER specific chaperone, is also increased the same way like HSP70, so can be used easily as a marker of hepatocellular carcinoma. And we have seen in our group with a retrospective series that effectively, if you compare non-tumoral with tumor in patients with different etiologies, this is ALCOL, this is MAFLD or MASLD, and you can see that there is increased expression of BIP in these tumors. We have seen this in different histological types, and we are now studying, Guillermo is studying the relationship between this increased ER stress and the immune environment. And then in the lab, Eva has looked to different mouse models of HCC. This is a transposon system that I'm going to talk about this today, but this is also with DEN plus CCL4, you can see increased amount of BIP, and then with the MDR2 knockout mice that develop also liver cancer, these tumors have increased amount of BIP. And then the question is, this BIP is only in the cancer cells, but or is also in the immune cells. So the response is not completely clear, but at least we can see we can say that cancer cells have a lot of stress. This is a human single cell HCC experiment, and we have looked these UPR targets. This is one of them. This is a chaperone. It's a XBP1 target, and you can see that it's increased precisely in the cancer cells, while other immune cells, these are memory CD4, macrophages, are not so abundant in these targets. So we think that UPR is important. It's important for the cancer cell to survive. And actually, this is data from the TCGA, and we have divided the tumors in the TCGA between P53 loss and beta-catenin mutant. This has been done by Sergio, and it is in most of the pathways that we are checking, there is a trend or a significant poorer survival in the patients that are overexpressing the signatures of these three transcription factors, indicating that the UPR is not only overexpressed or not only activated in the HCC, but this activation leads to poor prognosis. So why cancer cells need to sustain it and pro-survival UPR? Because increased survival against ER stressors like low glucose, hypoxia, so that they can modulate protein translation to avoid a prototoxic stress, so that they can promote efficient ER-associated degradation of misfolded proteins. And also another specific function of XBP1 this is during my thesis work. So we saw that this is the partial hepatectomy model of liver regeneration. So the 0 is the moment when you do the hepatectomy, and now we follow the mice, the colonies during all these hours, days, so that we can you are taking livers from. So we are sacrificing mice at different time points, so we can see the activation of different pathways. So we looked at the activation of the XBP1. This is the splicing of XBP1. You can see that very sharply after partial hepatectomy, it was increased, and also the targets like RAMP4 or HJ, they were also activated after partial hepatectomy. And since this was happening a little bit also in the sham mice, so in those mice were not operated, but they were open with a laparotomy, but they were not operated. So we thought this was a part of the acute phase response, and was exactly this way. And then when we took out XBP1, you can see that this is adeno shRNA vectors that are in vivo transfecting the livers, transducing the livers, and are depleting the livers of XBP1. As you can see in these mice, when you take out XBP1, they become more and more stressed. So you can see activation of PERK, you can see phosphorylation of AF2 alpha, you can see more translation of ATF4, of CHOP, you can see more cleavage of ATF6. So there is more stress. Logically, there is a lot of more stress in these livers, and there is more necrosis in these livers, and then these livers were these hepatocytes were not proliferating, and we did a ChIP-seq experiment to see in which regions XBP1 was binding, and this was the confirmation that the ChIP-seq was good. So it was binding. So XBP1 was binding all these chaperone genes that help the hepatocyte to fold better the proteins. But it was also binding some genes, some unexpected genes, which are part of the DNA response, DNA damage response. So it was very interesting, and we thought this was part of the reason why these livers couldn't regenerate properly. And this is an example of the genes. This is FEN1, is very important for repairing the DNA after DNA damage. And you can see in this SH-XBP1, so we are depleting XBP1 of these mice, of these hepatocytes. And we could see that this is a marker of double strand break in the DNA. So we could see increase of the DNA damage if we silence XBP1. So basically, in summary, we had this situation where during the regenerative stress, the replication stress, there were some important functions of XBP1, like acute phase response, UPR, but also DNA damage response. So and that after these results, we were thinking that probably XBP1 was very important for cancer, which is a cell that is completely continuously exposed to replicative stress. So basically, this overall hypothesis of our work is that if you have UPR activated, you can cope with the stress, and the HCC will pass unaverted and could evade the immune system very well. But if you inhibit the UPR, you could have higher ER stress, you could have higher TMB because you are impairing the repair of double strand breaks, and you could have also DAMs liberating for these dead cells, and then you can increase, the immunogenicity of these tumors. So to do that, remember these three transcription factors. We use a system that was kindly provided these plasmids by Amaya Lujambio in Memorial, and basically the system is used to generate tumors in vivo. And the way this works is that you inject these plasmids with high volume in an only one injection, in a continuous injection, which makes a situation of high venous pressure in the liver that dilates the endothelial system, and so the plasmids can enter in the hepatocytes without the need of any vector. So it is very low efficient, so only 2%, 5% of the cells can enter these plasmids, but just that those are enough to generate tumors, single or multiple. And then what we have done is that we have taken these tumors that are knocked out of p53 with MYC overexpression, or they can be beta-catenin mutated beta-catenin overexpression plus MYC overexpression. If you take out the MYC overexpression, you don't have tumor, so you need MYC overexpression plus any of these two human-like mutations. So we wanted something that resembles human hepatocellular carcinoma. This is why we use these two type of driver mutations. So as soon as we have the cancer cells, we take one clone and we again do CRISPR for each one of these transcription factors. So we generate a cancer cell that is from hepatocellular carcinoma, is syngenic with the mice, and is p53 or beta-catenin, and finally is knocked out for one of these three transcription factors. And then we use these cells for subcutaneous or orthotopic models. So this is just to show that this system works. So we have very well knocked out cells for our transcription factors, and we can see the targets that are completely reduced. This is BIP. And then we have done in the three transcription factors. So we are now investigating what is the role of these transcription factors in the immunogenicity. So we did this experiment with subcutaneous tumor model, and then we treat these mice with anti-PD-1 therapy, and then we sacrifice the mice at the end of the experiment or when there is a human endpoint. And as you can see, so in the cell line that we have created is not very sensitive to immunotherapy, but at least it has some sensitivity to the immunotherapy. The ATF6 knockout cells were equally sensitive than the controls to immunotherapy, but very surprisingly, we thought the contrary. But we found that XBP1 knockout made these cells completely resistant to the immune checkpoint inhibitor. So it was very, very surprising for us. As you can see, there is a difference between survival, between treating or not treating, treating or not treating. But in this case, we had low progressive cancer, but as soon as it develops, it kills the cells, it kills the animals, even if we are treating with immunotherapy. So basically, we were wondering what was the mechanism, and we are investigating it and completing this description right now. So we thought that maybe XBP1 was important for the antigen presentation machinery, because the antigen presentation machinery, which is the system the immune system uses to detect endogenous and exogenous antigens and kill the cells through the interaction between the TCRs and the peptide load MHC class 1. Everything starts in the endoplasmic reticulum. So the peptides that are degraded with a specific pattern in the proteasome have to be translocated to the ER, and the ER is when they load the MHC class 1 that is already combined with a beta-2 macroglobulin. And so all this system has to be very well regulated. And we thought that maybe XBP1 has to do with this. So if you don't have XBP1, maybe you are not showing the tumoral antigens to the membrane. So we tested this hypothesis. Before that, we saw that if you treat with interferon, which is a molecule that elicits the MHC class 1 presentation in the membrane, we also activated the UPR. So actually, this is 24 and 48 hours. So you have an increase of XBP1. At 48, you had an increase of ATF6. And with ETF4, it's not so clear. It seems that there is an increase at the beginning and then rapidly decrease after 48 hours. So we use this system to see if treating the cancer cell with interferon, so inducing the expression of the translocation to the membrane of the MHC class 1, so we could see them in the flow cytometry, and we can measure the amount of MHC class 1 exposed to the membrane. And this is what we did. First, we did with a simple experiment with HCC cell lines P53 mutated, but were wild type for the UPR. And we just did a treatment with tunicamycin. Tunicamycin is a drug that produces ER stress because it blocks the glycosylation of proteins. So we saw how there was dose-dependent inhibition of the exposition of the MHC class 1. So it was interesting. It means that when the cell is stressed, it exposes less the MHC class 1. Maybe it cannot be folded. Maybe there is a degradation of the MHC class 1. We don't know. And then we did the same with the knockout cells. So we saw that especially at 48 hours, there is a decrease on the exposition of MHC class 1 in the XBP1 knockout cell line. So we think that this could be one of the mechanisms why these tumoral cells are not responding to anti-PD-1 therapy. And we also saw that many of the targets that are needed for the proper folding of the MHC class 1 and the loading with the antigen, with the peptide antigens, were also downregulated in the XBP1 knockout cells. So to conclude, we have seen that the HCC is polyimmunogenic or is immunosuppressive. So we need strategies to make them more immunogenic. We have seen that endoplasmic reticular stress and the photoapoptotic response are potential targets for improved systemic treatments in this disease. Our experimental setting, as I have shown you, allows for the integration of single targets in a syngeneic union-like immune-competent model of HCC. And then I have shown these preliminary results of the necessity of XBP1 by the cell to do a proper exposition of MHC class 1 and be identified by the effector immune cells. And that could be a mechanism of lack of immunotherapy efficacy. And now we are doing a lot of other work, interrogating the other arms of the UPR and confirming these results with beta-catenin mutated cells and other projects ongoing. So are we happy? So thanks to all this work, I'm working 3 days in the clinic, 2 days in the lab, and these are the ones that are working all day with these experiments. There are also other collaborators. Stefan is coming from Peru. Guillermo is a pathologist, is now in Belfast, but he keeps collaborating with us with identification on biomarkers of ER. Alex is a medical student, and is working with single cell sequencing analysis. Miguel is a resident, is working in different projects also related with HCC immunogenicity. This is the whole team of the liver unit in CUN. You know some of them probably. And there's lots of people collaborating with us, including in the BCLC and in the Gregorio Marañón. So thank you very much, and I'm open to any question you can have. were very focused on the immune synapse, so the interaction with the immune system, so the immunogenicity in vivo and in vitro. But this is something that we have to do. Yeah, that's a very good question. So we have to see if these cells are actually having more DNA damage and maybe more mutation burden in vitro. So that will be very interesting to see. Thank you for the suggestion. So I think this is, I don't know, but I think it's a very good question. And the difficulty of looking at the ER stress and the unfolded protein response is that we don't have markers to distinguish between a cell that has overexpression of the UPR elements or a cell that is stressed. So a cell that is very well coping to the environment, so it has an overregulation of the UPR system, versus a cell that is very stressed, so it has a lot of UPR activation. So we cannot distinguish between these two. I will say that NASH or MAFLD or MASLD has more ER stress. That has been shown 20 years ago by Otamisli Hill. The first evidence on liver ER stress was in a diabetic cohort, so it was MASLD. Whereas in alcohol, there are different phenotypes, and I haven't seen. There are some works by a group in California on alcohol and ER stress, but I haven't seen stronger, equally strong data, I will say. But I don't know about different patterns of activation. This is a very interesting question. So first of all, I will say that we don't know if as you have seen in the J hepatology in 2003, so it was different differentiations. You had different stress situations. So maybe also in evolving, for example, a larger tumor that has more hypoxia, the environment is completely different, but also the stress could be different. So maybe the efficacy of the treatment that is targeting the UPR could be different. So this is something. It's very soon to talk about that, but this is very important to take into account. And then for relating to that's a very important question. This is why I'm working with a single cell sequencing data to see which type of cells are activating which type of UPR arms. So we don't have. So we need better single cell sequencing, deeper single cell sequencing to see, the genes that are low expressed, but they are markers of a specific pathway within the UPR. But for what I can say that we have cancer cells that have UPR, but also we have macrophages that have UPR. So in both cell types, if you do a systemic treatment, you could damper the UPR response in both cell types and maybe having different effects. So this is something that has to be investigated. It's very, very interesting and very exciting. So I think that the clinical trials, so now the industry is leading the phase 3 clinical trials with a lot of patients, with a lot of sampling potential, potential for sampling, hundreds of patients that are treating with the same drug, and I very well follow up. So I think these main, the main companies right now are doing spatial transcriptomic analysis on paraffin fixed tissue. This is a first step. I don't think we can do single cell for all these patients that are entering. It's very difficult logistically. They are not going to spend all this money if there is not something already there. But at least I think in the following years, we're going to see spatial transcriptomic data of patients that are responding or not responding, which is the specific type of immune environment in these cancers. But I think personalizing cancer therapy will be it needs to be this way, no? So, you know, I don't know how far we are, but.
Thank you very much, Jose Maria, for this excellent talk, for accepting the invitation, and all the attendees that have been with us today. And thank you very much for the opportunity for being the host today, and hope to see you soon.
Thank you, Anna.